everybody. Yeah, so today I'll be talking about the role of emergence in open-ended systems. And I'm really excited to be here because this is a accumulation of work that I've been doing over the past 5 years. So from my PhD work and into my current position right now. So basically, I want to talk about my background first, because if I talk about my background, it sort of makes what I study make a lot more sense. So I have a sort of diverse educational background. So I started off in physics with my bachelor's degree in 2013. And then for graduate school, I studied with Sarah Walker. And even though it says physics on my degree, I actually studied what the difference between living systems and non-living systems are. So it's a little bit non-traditional in terms of like what a physics PhD is. It's more like astrobiology is what I studied. But I specifically looked at the role of open-ended evolution and innovation in biological evolving systems. And I also looked at discrete finite deterministic systems, which are cellular automata, which is why we are all here. In 2017, I also did a internship at Microsoft, and I learned a little bit about machine learning. And then I went into industry for a couple of years as a data scientist, where I learned about Python development and software development, problems with big data and things like that. But I found that experience to be extremely valuable because a lot of the things that I study involve a lot of data, especially when it comes to biological data. And then after spending 2 years in data science, I decided to return to academia to work on these problems full-time instead of part-time. And I've been there since last October, and I'm working in bacteriology, bioinformatics, where I look at genetic sequences and protein sequences of bacteria and viruses. And I'm currently interested in the emergence of this idea of function as well. So when you have all these microbial communities work together and they all do a similar thing, what is the computational mechanisms behind that? That's what I'm interested in. So a little bit more specific, and hopefully these questions that make sense, these questions that I study make a little bit more sense because of my background. So I'm still really interested in this idea of how are living systems different than non-living ones. So I'm asking a little bit more specifically is what are the unique mechanisms of living systems? And this is markedly different than just thinking about what is alive, because a lot of the times the discussion about what makes something alive deals with metabolism, reproduction, and things like that. But I'm thinking more of a living system as a whole. So if I'm thinking about ants, I'm including the anthill, and if I'm thinking about human living systems, I'm also including the buildings and human technology and things like that, which is also known as signatures of life as well. So system as a whole. And I also think about, can statistical mechanics be useful for biology in any way? So there's a lot of relationships between different levels of organization in biology, and I want to know if statistical mechanics can help inform about how those levels of organization interact with each other. And if we think about things in different levels of organization, can we understand emergence better that way? So, and I also study how does life arrange itself in physical space to perform computation. So in biology, it's well known that structure equals function, and I want to think about that in a formalized way in terms of computation. And then of course, what can real biological data tell us? So if I have a bunch of genetic sequences of viruses, bacteria, and I also have a lot of data from Twitter, from human social systems, and data that I can find from the internet, how can I utilize all of this vast amounts of data to inform us about the underlying mechanisms of how biology works? So a little bit more visually and a little bit more silly, I study some sort of intersection between bioinformatics, information, computation, statistical physics, and biological evolution. I have a lot of background in different fields of study as well. So if you have a question, if I say something that doesn't make sense, because oftentimes going from one field to another field, I'll say a word and that word means different things in different fields, so it doesn't sound like it makes any sense. Please ask me any questions that you might have, because I'm always happy to ask questions and to clarify things going from field to field. But otherwise, if I'm not asked to clarify things, I can start to look very crazy very quickly. So, all right, now it's time for 3 stories. So the first story is about 30,000 base pairs. They arrange themselves in some particular order, and as a result, most of Earth's humans suddenly stay inside small structures more frequently than they would otherwise. And another time, Elon Musk posts an arrangement of letters on Twitter, and as a result, the stock market takes a hit. Another story is a butterfly flaps its wings in the Amazon, and then you get tornadoes in Texas. So typically, when we hear about these examples, they're associated with chaos theory. But actually, in biology, that's not really what always happens. More realistically, things aren't so causally exciting. More realistically, if you have 30,000 base pairs arranged in some particular order, and if it's a virus, then it could be malformed and it does nothing, which is what viruses are actually pretty good at, is deforming themselves. So another realistic scenario is that I post an arrangement of letters on Twitter and I get 2 likes, and that's that. Another realistic scenario is a butterfly flaps its wings in the Amazon and its neighbor is graced with a refreshing breeze. So like I said, this usually talks about chaos theory, but I'm going to talk about these in terms of emergence. So emergence is a little bit different than chaos theory, because with chaos theory, you're talking about sensitivity to initial conditions and how we're able to make predictions from a perturbation. So if we make a small change in the system, how does that affect how the system is behaving overall? Emergence is an idea that is most commonly understood with this famous quote by Aristotle, that the whole is different than the sum of the parts. So now most of the times it's misquoted as the whole is more than the sum of its parts, but it's actually different than the sum of its parts. So with these examples, you have a change that's happening on some level of description in a system, but as a result, you get some dynamics that happen on a different level of description in that system. There are 2 kinds of notions to emergence, because the word gets thrown around in many different contexts. Well, maybe thrown around is a bad way to say it, but it gets used in several different contexts and they all mean different things. And the way that I think about emergence is in 2 forms, which is strong emergence and weak emergence. And there's this nice paper by Marc Bedeau from 1997 that talks about the difference between weak emergence and strong emergence. So I highly recommend you read it. But for weak emergence, that's kind of the more colloquial idea of what emergence is. So sort of like the cat emerged from under the bed, or Ebola, the disease, emerged once again. So it's basically the appearance of something new from a known possibility space. So it's expanding a known state space. So if I'm thinking about words, I have a word state space and a new word might emerge. So that's an idea of weak emergence, because I took my word state space, I added a new word, and my state space expanded. Now, this idea of strong emergence goes further than that. So strong emergence examples are like the emergence of multicellularity in biological evolution, or perhaps the emergence of language in human behavior. So it's not just the expanding a known state space, it's actually the appearance of an entirely new possibility space. So if I'm thinking about my word state space as an example, that's one state space, but you get another state space from it called the story state space. We put words together to create stories together. Also related to this idea of strong emergence is coarse graining, and they're a little bit different, so I think it's worth noting. So coarse graining is when you condense a state space into a smaller one. So for words, if I were to take a bunch of like maybe 5 words, and now I say that they mean the same thing, and I can just condense them into a single word, that's what I'm doing. So I'm taking a state space and I'm making it smaller. So that results in a many-to-one map. It says one-to-many maps, but that's a mistake. So it's many-to-one maps, which is also loss of information. And it also has a really nice result in the world of cellular automata. So if you have a regular elementary cellular automata with your periodic boundary conditions, very classic CA experiment, and if you coarse grain all of the different states from all the possible rules and all the possible initial conditions, and you coarse grain that into a smaller CA, what happens is you can say, like, what states do I get in that coarse grain CA? Do I get something that looks like a CA that's following one of the 256 elementary cellular automata rules? And the answer is yes. And when you do, you actually decrease in complexity according to your Wolfram complexity classification scheme. So if you have something that's really, really complex, that's your microstate, so it's a larger CA, and if you coarse grain it into a smaller CA, then you can only go down or stay the same in complexity. But I don't necessarily think that's a bad thing. I suspect that biology might utilize coarse graining as a way to make computation easier, because I think maybe it could be possible if you have a large state space, it could be more difficult to navigate, especially if it's more complex. So if you have something that's easier and less complex, maybe it's something that biology uses. I don't know. I don't I don't address that question specifically, but I think it's definitely worth thinking about for sure. So I talked about emergence, and then the other part of the title of my talk is this idea of open-endedness. So there's this really nice article in 2017 by Kenneth Stanley and Lehman and Soros, and it talks about why open-endedness is such an interesting problem, and specifically in this case, why it relates to artificial intelligence and machine learning. So currently, in the context of machine learning, these systems are really good at solving a known problem, right? So they find and they do some sort of like novelty search to look through all these different solutions, and then it finds and picks the best one. But that's different than what biology does, because what biology does is it not only solves a problem by looking through different solutions, but it creates new problems to solve. And then that is what open-endedness is. So you cannot get open-endedness from machine learning systems simply because they're not coming up with new problems to solve, they're only solving a known problem. So the idea in terms of maybe for instead of machine learning, if we go to biology, we think of biological evolution as only going forwards over time. It doesn't repeat itself over time. And instead, it just constantly finds new novel solutions to problems while also creating new problems to solve as well. So more biology, more problems. So definitions of open-ended evolution. So a little bit more formally, because that was all a whole bunch of formal language to describe what open-endedness is. There's a lot of discussion on how to formalize this for discrete computational systems. So there's still, I think, a lack of generalized consensus about a strong definition of open-endedness, but there are some things that people can agree on anyways. So these are 4 things that people agree on what open-endedness is, and it's the ongoing innovation and generation of novelty over time, so sort of like the biological fossil record, something like that. It's also unbounded, meaning it doesn't repeat itself when you expect it to. And it's constantly producing complexity over time, just like biological systems, and it's a defining feature of life. So what does this exactly look like in finite deterministic bounded systems like cellular automata? So the problem with cellular automata is that they're bounded in many ways by this Poincaré recurrence theorem. So that says that the state of every finite deterministic system approaches the initial condition within a sufficient amount of time. So for elementary cellular automata, if you get back to a state under a fixed rule, you're only going to repeat yourself because it's a fixed rule and you give it a state and it's totally deterministic and you have no choice but to repeat yourself. So this gives it a natural bound on complexity, it gives a natural bound on the number of possible states that a system can look at, and it also gives a natural bound on open-endedness, because if you're always just repeating yourself, there's no notion of innovation whatsoever. So what would it take in order for a system to be open-ended, especially for cellular automata? What would the cellular automata have to do for this to be open-ended? So it would have to do 2 things. It would have to be innovative, and it would also have to be unbounded as well. So the idea of innovation for a cellular automata system is basically it's taking a unique path through the state space. So for ECA, it's really easy to enumerate all your possible trajectories through a state space because you just have a finite number of them. But what it would take to be innovative is if a CA were to take a path that was not seen before. So that depends on counterfactuals from just having a regular cellular automata system that's isolated from everything else, because that's what we already know. So for unbounded evolution, it's not just taking a unique path through state space, but it's whatever path that you take is going to take longer than the Poincaré recurrence time to start repeating itself. So when it's going to reach its attractor cycle or its attractor state, it takes longer, it takes more steps than the Poincaré recurrence time for an ECA. So all of these are completely impossible for regular ECA by definition. I will just say exactly the previous slide before, but in a visual representation. So there are 4 parts to this figure. There's A, B, C, and D. So for A and B, these are just regular elementary cellular automata that everybody knows. These are state transition diagrams. So on my nodes, they are states, and for this particular CA, there's only 6 possible states. And I have rule alpha for part A, and then rule beta for part B. So for under rule alpha, if I start in, for example, state 3, I'm only going to go from 3 to 5 to 3 to 5, 3 to 5, and then that's my attractor cycle forever. But of course, I have a different rule. My topology of my state transition diagram is going to look different. So if I started in state 3, I'm only just going to do beta over, or if I started in state 3, according to rule beta, I'm just going to do 3 over and over again, and that's my attractor state. So innovation, what that means is if I were to look at all my possible initial conditions and all my possible rules, I get a set of possible state trajectories that I can go through. But innovation means that I take a different trajectory. But what that needs to include is it means that I'm just not looking at rule alpha or just rule beta. I'm looking at multiple rules at the same time, because that's the only way that I'm able to get a different topology. So if I'm a CA and I am going to use rule alpha and rule beta, what that means is that if I start at, for example, state 3, I can use rule alpha, and then if I'm in 5, well, I can go to state 6 by using beta. So I get a unique trajectory through the state space. Now, unbounded evolution means that not only do I get a unique path through the state space, but the time that it takes for me to come into my attractor cycle takes way longer than it would for regular ECA. So just to recap, because that was a lot, this is all impossible for regular elementary cellular automata. But if I were to have open-endedness in a CA system that uses ECA rules, then I have to be able to change my rules over time. So I'm going to be using a coupled CA system to do this, where there are one CA and another CA that are spatially separate, and they don't share any boundaries or any cells or anything like that, but they still interact in a particular way. Basically, what all this is if we go back to our states of our different state spaces, open-endedness is only looking, it describes the behavior of how you move throughout a particular state space. Whereas innovation, remember, is about it's either expanding your current state space or it's creating an entirely new state space altogether. That's how those two relate to each other. So emergence is creating new state spaces and expanding a known state space, where open-endedness is how you move through the state space. And biology seems to do both, which is why I'm really interested in studying it. So, okay, why do I have 2 separate CAs? Let's go back and start thinking about biology again, because everything that I want to model, I want to be inspired by biology, so it can tell me something back about biology. So state spaces, biological state spaces, do not exist in isolation whatsoever. So it's sort of like this problem, this old puzzle that you might see from elementary school, is how many triangles do you see, right? So you have this one big triangle and you have lots of like little small triangles, but you also have the overlapping triangles, and you also have some upside down triangles, or, well, whatever, you know, if you consider this right side up. So you have lots and lots of different possible triangles here. That's not so different than looking at a picture like this, right? So we look at living systems in an environment, and there's all sorts of different state spaces that are overlapping with each other. So there's things like temperature, there's things like pH, there's things like taxonomy and population and individuals and evolution. So we humans pick particular state spaces to describe biology that allows us to do computation and make predictions about what's in the future. So it's really important for us to understand how these state spaces overlap with each other and interact with each other. That's why I have a coupled CA. So if I have a coupled CA, that means that I can assume that, well, I have one partition of biology, and then I also have another partition of biology. How do those 2 state spaces interact with each other? So that's why I have that kind of coupled CA system like that. So this is a visual representation of what the model looks like. I have a environment cellular automata and an organism cellular automata. And they're both completely spatially separate. And the environment is going to just evolve like a regular elementary cellular automata with periodic boundary conditions. However, my organism cellular automata is not going to have a static rule like an ECA. Instead, that organism is going to be able to change its update rules over time. So I studied this. This was the model that I studied as a part of my dissertation. And we studied how these two CA could interact with each other in a few different ways. The organism could change its update rule based off of its current state and also the state of the environment. Which, it's basically just saying like, okay, give me a mapping between the state of the current CA for the organism and the state of the environment, and just give me some sort of static mapping between those two things in a rule. For this part, we basically just picked a mapping. It's a static mapping. It doesn't change. It's also just a completely arbitrary mapping as well. There's nothing really special about it is what I'm trying to say. But we also thought, okay, what if the organism doesn't necessarily use its current state to update its rule, right? Because if it's using its current state to update its own rule, it's using what's known as self-reference. And biology is always using self-reference because you always have to use your current state to get to where you're at next. But we thought, well, what would happen if we just threw that part away? So instead of using its current state, it's just using the state of the environment. So you can think of it as a totally open system. It's completely open to the environment as a driving force to change its update rule. And then finally, we wanted to explore this idea of randomness as well. So rather than having the rule change based on states, we just had it change randomly. So this is not a static state to rule mapping. And as a result, what we found is that for the case that it uses self-reference to change its update rule, we got a lot more open-ended cases. And we also found that as the environment grows larger over time, the more that you get open-ended cases as well in your organism, which makes a lot of sense. It makes sense in terms of biology, but it also makes sense in terms of physics when you think of, say, coupled oscillators. So as your CA are both evolving over time, if they're in their attractor cycles, they're basically like some sort of oscillator. So if you have a larger environment, then you're going to get larger attractor cycles. So you have a larger, I guess you have like a larger driving force than your organism. Or maybe it's not larger, but it just has a longer periodicity. We also wanted to know more about the topologies of what these state space mappings look like. So for regular ECA, again, you can just make these graphs saying which state evolves to which other state over time. And of course, according to your different rules, you're going to get different topologies. We were also interested in this idea of physical universality as well. Physical universality is really interesting for biology because it has to do with like the things that biology can't do, right? So it's saying that if I'm in a particular state and I'm a biological system, physical universality means I can get to any other state. So biology is actually pretty good at this, although we don't really know how physical universal biology is, because can it get to any state? It's not something that we're able to that we're not really sure about right now. My advisor had a really, really, really great example that talks about it. Before biology, Earth only had one satellite, which is the moon. But then after biology, now Earth has a ton of satellites, thousands, 10,000, I'm not really sure. But the point is that biology was able to make a change in the physical universe that was previously impossible without biology. If we're thinking about these discrete dynamical systems, we're thinking about like, how do we go from one state to any other state? So if a system is fully physically universal, that means no matter what state I'm in, I just have to wait a particular amount of time and then I'll get into another state after that. If you look at these state transition diagrams, clearly, if you're stuck in one of these islands, you're never going to get to another island over there. So we were really interested in, do open-ended cellular automata, do they look like they have physical universality? And is there a relationship between physical universality and open-endedness? a quick sort of like reminder of physical universality, it's really, we're thinking about Janzing's definition of physical universality in a discrete system, but we modify that definition slightly to being locally physically universal. So rather than thinking about all possible states that a system can have, we're just thinking about all of the accessible states. So it's a little bit different. It's not exactly true physical universality, but it's local physical universality. So here's some examples of what those topology networks look like. For A, this RFP value is 0. So that means locally physically universal, it's not, it's 0. So every single state that you're in, you can only get to a attractor state, and then that's it. So that's basically the equivalent of I have reached the state of all zeros or all ones, and I'm never going to go anywhere again. That's opposite of physically universal. So, but if you have something that is fully physically universal, then this value would be 1. So R sub P would be equal to 1. So these are all state transition diagrams from that coupled system that is capable of open-ended evolution. And you could see that there's a lot of variability in terms of like the different locally physically universality. So if you have a topology that has a really high R sub P value, you also get a lot of open-ended cases. It turns out that, yeah, you do get open-ended cases, but not as many as at least I expected anyways. These are different sizes of CA. So W equals 5 means it's only 5 cells wide, and of course there's still periodic boundary conditions. So on your x-axis, you have different R sub P values. So over here, you have 100. These are in percent. This is my mistake as a grad student. Instead of having 1 to 0, I did 0 to 100%. But you have 100% fully locally physically universal, and you do get a lot of open-ended cases here. So this is also a distribution of the fraction of your recurrence time that it takes for you to reach an attractor, divided by the Poincaré recurrence time. So basically anything above the 1 on the y-axis is going to be open-ended. So anything that's above that line is an open-ended case. So now I'm thinking of, how does biology create new state spaces? Or in other words, what are the mechanisms of strong emergence? Well, how does that happen? And how does biology create new states, which is basically asking, what are the mechanisms of weak emergence? And also, how do state spaces interact with each other? And what gives a state space more predictive power than another state space, according to a particular observer? So in other words, for us humans, why is it useful for us to think about the world in terms of chemistry on a particular scale versus thinking about it in terms of population dynamics on a different scale? You know, what makes that so strong? And does all of this have a relationship with complexity as well? And of course, how can we use real biological data to inform these kinds of models? So for the next part that I did is I took the same setup that I had, but I modified it a little bit because I'm thinking more about these state spaces. You know, if we have the current positions and velocities of a bunch of birds, then can we predict the shape of a flock in the future? And if we have the neural activity of a brain, can we predict maybe like how a person is feeling or what they're thinking or something like that? Or if we have what everybody's talking about in 1788, can we predict the exact day of the French Revolution that happens in the next year? Or if we have a chemical soup, can we predict if and when life will emerge? And if we have a genetic code for a virus, can we predict how it's going to evolve over the next few years? So this is really getting at this idea of how are these different state spaces interacting with each other. It's not necessarily chaos theory or like the other example, but it's rethinking about that same problem in a different way. Is if we're thinking about things on one state, how does it affect it? How does it affect our system in a different state? This is what so for this next part, I study this question of how do different state spaces interact with each other. So for the coupled CA, we have the state of our environment, and then we also have the state of our organism, but we also have the current state of the rules in both, and we also have the state of the entire thing, but we also have the state of like the rule mapping between the states and the rules. So there's all kinds of different states that are going on here, state spaces that we can study. Because biology, we found that we get a lot of open-ended cases using self-reference. Instead of having this setup with your environment and your organism, I next looked at this setup where you have two CA that are constantly changing their update rule rather than just one CA changing their update rule. So just like the other part, I looked at different ways that these two things can interact with each other. So the first way is that both CA are going to use the CA of the entire system, so both CA. So in order to calculate the next update rule, they're looking at both states as a function of that. Or if I'm a CA, I can use my current state to update my next rule. Or I can use the state of the other CA as my update rule. So either I'm using my own state only, or I'm using the other state only. Or it could be that I have a bunch of states and I have a random state dependency. So either when I'm determining my next update rule, I make a random choice of, am I going to use both states, my state, or the other state? So I did the same experiment again, but if you notice, I'm not doing this thing again because this is just one static mapping between states and rules. But now that I have different kinds of states, I'm going to be thinking about it in terms of networks instead. I create all of these random static mappings, which is like a bipartite map between states and rules. So either it's the combined state of the both CAs, or it's just a single CA, and that's just a static mapping to some rules that CA can use to update themselves next. So the questions that I'm thinking about with this, because I can make whatever mapping I want. I generate a whole bunch of them, and I'm going to ask, if I have a mapping that's more complex, does that lead to more complex state space trajectories? Or does it lead to open-endedness? So this was just a preliminary experiment that I did. So I generated 5,000 random mappings between the states and the rules. I turned this bipartite graph into an adjacency matrix. And then I used the block decomposition method to approximate the algorithmic complexity, which is also the Kolmogorov complexity as well. But because these mappings were generated randomly, I'm going to get a low VDM value for each, so it's a low complexity value. So they're going to have about the same values. So if I'm thinking about like more complex state rule mappings versus less complex state rule mappings, then I have to do it relatively. All of those 5,000, I picked like the highest complex ones and the lowest complex ones. So those are my methods. And also because it's really computationally expensive, I only looked at width of 3 cells only. So these are really, really tiny CA. So as a result, what I noticed was that I got the most interesting cases for when the state rule mappings was a random choice between my state, the other state, or the both states if I'm a CA. So for the first figure on the left, what I'm looking at is on the x-axis, I have the recurrence times, and this is a frequency distribution plot. So it's basically just a histogram, like what sort of recurrence times do I get out of this? So for the random case, I get a lot more cases that are unbounded. So they're not necessarily innovative as well. So they're not necessarily open-ended, but they are unbounded in the sense that they don't reach their attractor within the Poincaré recurrence time. So, and then for the other plot, the one on the right, we have 4 panels, and these are for the 4 different cases. So, and this is the complexity of the average complexity of these states. That's your x-axis is your average complexity, and then your y-axis is your frequency, because these are also all histograms as well. So you don't see like a super strong relationship for all the cases, except for the random one, which is the bottom right-hand corner. So you get this really interesting bimodal distribution of certain complexity values, which is really interesting. So these are just preliminary results. To answer our questions that we were thinking about is, do more complex state rule mappings lead to more complex state space trajectories? And no, there wasn't a relationship there. So, and does it lead to more open-ended evolution? And no, there was also not a relationship there too. So I had plots, some scatter plots, but there wasn't a relationship, and there also wasn't that many points. So I'm just going to tell you about it instead. But what we did see is that if you randomly change the mapping type between the rules and the states, meaning I make a random choice between how I'm going to, or like I make a random choice about my state dependency when it comes to choosing my rule, I get more open-ended cases, and I also get more complex states and more complex rule trajectories as well as I'm going through the different rules. So that's pretty interesting, but I also have a ton of follow-up questions, of course. I would like to do the same experiment, but with a better range of mappings between states and rules, because, you know, at first I just randomly made them, so they're all going to have a really low complexity value. I need more mappings that are diverse in terms of BDM. But I also want to explore this idea of one-to-many maps, one-to-one maps, and many-to-one maps as well between states and rules. And also, how else can I implement these different mappings where I'm losing information? And how does that loss of information contribute to this idea of open-endedness? And of course, I want to do it with larger CA of different sizes. So maybe one CA is larger than the other one or something like that. And it also, rather than just having two CA that interact with each other, what if I had more? What if I had like 5? What would happen then? So this is all totally a work in progress. So always happy to chat more if you have ideas. But you can see my preprint. I just submitted it to the archive server, so you can view it there as well. So I still have all of these specific questions that I study. I still want to know what's the relationship with complexity between models with interacting states, and how can I use real biological data to inform these models. So not only am I looking at these questions in terms of modeling with CA and also things like Ising models and mathematical models in general, but I'm also thinking about these questions in terms of real biology, because I want my results from real biological systems to inform how I'm making these models, and then they can inform each other. One of the projects that I'm working on is this idea of, rather than having a CA and a CA interact with each other, I'm thinking about how viruses interact with hosts that they infect. So this isn't a state space topology, but what this is it's a protein-protein interaction network. So your red proteins are the proteins from the virus of coronavirus, which is what everybody's dealing with right now. And then all of the other colored nodes are human proteins. So this shows which COVID proteins interact with which human proteins. And with these proteins, you can measure the BDM of them as well. So I want to know, like, do complex proteins, do they do anything that's special that in comparison with non-complex proteins, or like what's the relationship between complexity and something interacting? I asked for the data from the authors from this paper. So like this paper is from UCSF, it's a preprint, and I asked them for the sequences for both the for the coronavirus, and then I also got the human sequences from Unipro. And I'm going to use BDM, which of course it was the same BDM that I used before as an approximation to algorithmic complexity. But I'm also going to, rather than just look at the complexity of the entire sequence, I'm going to look at the complexity along the length of the sequence as well. So I'm really interested in, like, are there areas of high complexity or low complexity? And then I was also just being silly and messing around, and like if you look at the sliding window, you get a certain value, and if you bin those values, you can into discrete values, and you can measure the BDM of that sequence's values as well. And I also did it a third time, but it doesn't actually mean anything. So it's nothing significant. I was just messing around. So the questions that I'm asking are, for COVID, are these proteins interacting with other human proteins with similar BDM values? And do viral proteins with the same function, do they have the same BDM values as well? So it turns out that if I were to look at all of these different measurements of BDM, and I wanted to see, like, what's the relationship, it turns out that we actually do get a really strong relationship between highly complex proteins in COVID. They're very likely to interact with human proteins over ones of lower complexity. So complex proteins interact with complex proteins. So that's the main result is the last panel that's circled. All of those, the other 3 panels, you can just ignore, because those are like the silly things that I was messing around with just to see if I could see anything, which I didn't. But the on the x-axis, you have your viral protein BDM. That's your complexity value. So if it's close to 1, it means it's really complex. And same with the y-axis, but that's for the host. So those are your human proteins. And each dot represents a protein-protein interaction. All your gray dots are the ones that are not actually proteins that interact with each other, but I use them as a control to get to see if my result was significant or not. And also, going back to the other question of what's so special about complex regions of a protein. This is not coronavirus stuff. This is a bacteriophage, which is a virus that attacks bacteria. It's called T7. It's really famous and really well studied because it's a virus of E. coli, which is, of course, really well studied. And I got these really nice viral sequences of just the part of the virus that attaches to the host. And those sequences were mutated a whole lot. And so if I looked at all these different mutations of the attachment part, can I say like, okay, these, the regions with mutation have a high or a low complexity, or like what's the relationship between the two? So it turns out for conserved regions, the parts that don't mutate, they generally have a higher complexity value than the other parts. So the parts that mutate a lot have a really high spread in terms of what their complexity values look like. So they're BDM, which is really interesting. So to wrap up, what we know so far about how this looks in biological systems from real biological data is that regions of mutation within some viruses, which is just T7, they have lower complexity values. So is this true for every virus? Is this true for COVID? But for COVID, only the most complex proteins in COVID-19 interact with human proteins. And also, is this true for viruses in general? So I'm looking at all the experimentally confirmed protein-protein interactions between viruses and hosts, and I will redo this analysis across all known and experimentally verified interactions. So in other words, the adventure continues. So this was all preliminary results. This is not a, I don't have a preprint for this yet because we're still looking at all of the data, but I hope to have it soon. And so finally, I want to say thank you to my advisor, Kartik, and to the grad student in the lab, or to one of the grad students in the lab, Chris, who is really amazing. They're both really great. And of course, thanks to Hector, who has always been a really great mentor and works with me with BDM and is always really great to ask questions. And of course, to UCSF, the folks who helped me out and to get the COVID protein sequences. And then of course, thanks to everybody that worked with me in grad school and was super great. And of course, thank you to everybody in the conference, and thanks to the conference organizers for hosting and inviting me. And it's been a really great conference and I'm super excited. But before we go, I have one last thing for you. These are really pretty CA pictures and nobody appreciates them except for the CA community. These are the organisms that change their rule over time as a function of their own state and the state of a environment, which is not pictured, and they look beautiful. So I also think they would make lovely ties. So if you're interested in maybe coming up with some cool designs, you can contact me. So thank you so much for your attention, and I hope you all have a great day.